Hi everyone. In this episode we're going to use DataWarrior to do core-based SAR analysis. We'll take a dataset, define a common scaffold, the core, map R-groups, and read activity trends to suggest new compound designs. By the end, you'll know how to extract a core, build an R-group table, and generate design hypotheses all inside DataWarrior. We are going to look at structure-activity relationships, or SAR, and more specifically at core-based SAR. Instead of comparing entire molecules, we focus on the central scaffold, which is often named the core. We will search molecules in the ChEMBL database, so to start with, we will first draw a fragment in the chemical editor window. We draw the fragment shown in the figure, and then use the lasso tool and select the atoms in the ring as shown. Then we double-click on an atom and open the atom query features and select any atomic number. Then we double-click on a bond and open the bond query features and select single and double bonds. With this command, you should get over 100,000 compounds. Then we will keep only molecules interacting with the phosphodiesterase 5A, and for organism, we will select molecules active on Homo sapiens. We should have about 487 molecules. We can save these molecules just in case. The easiest is to save the file in the default DataWarrior format. Now that we've loaded the dataset, the first step is to sort the compounds based on their p-value. To do this, simply click on the p-value column header. DataWarrior will automatically sort the molecules in ascending or descending order. Next, we'll remove all compounds that have a p-value of 0, since these don't provide meaningful biological activity data and would introduce noise into our SAR analysis. Now, let's refine our dataset further by keeping only the molecules that have reported IC50 values. After applying this filter, we're left with around 347 compounds. Next, we'll clean up the table by deleting invisible rows. Then we can delete many columns that we are not needed here, select the ones you see on the screen, and save the file. Now we can start exploring the molecules. To do this, go to the Chemistry menu and select Analyze Scaffolds. DataWarrior will then identify the ring systems present in each molecule. We sort on p-value to have the best compound on top. Then we click on this best compound to copy the structure in 2D as shown. And then now we go to Chemistry and decompose R-groups. In the graphic window, we paste the 2D structure of the compound we selected in the previous slide, and with the graphic window we are going to select only a small part of the compound as shown on the screen. Then, as before, in this structure editor window, click on the atom, select any atomic number in the small window, and do the same for bonds. Select single and double bonds. You should have something similar to what you see on the screen. Several new columns are created. One column contains a scaffold for each compound, while the other columns correspond to the different R groups attached to that scaffold. At this stage, we're just getting familiar with DataWarrior's functionalities and understanding how the software organizes our data before we dive deeper into analyzing activity trends. We will now delete all these new created columns, and we will keep only some columns as shown, and then you can draw a small fragment in the structure editor filter as shown on the screen. You should have around 43 molecules. You delete all the others using the menu data and then delete invisible rows. We can then compute the scale sphere fingerprints from the Chemistry menu to remove duplicates. While this is not perfect, you can do compound clustering. You go to the Chemistry menu and select Cluster Compound using the scale sphere fingerprints and the similarity value of 0.99 as seen in the figure. Now, we'll reduce redundancy by keeping only one representative molecule per cluster. To do this, select Is Representative as shown on the screen. Next, go to the Data menu and choose Delete Invisible Rows. This removes all non-representative molecules, leaving us with a clean data set of 34 compounds, one representative structure from each cluster. And now in Chemistry, we do again decompose R-groups. You generate several new columns with the core scaffold and the R-group columns. We can then again sort on the p-values, so we have the best compound from this dataset on top. Now we can examine the molecules to see which ones have strong inhibition values and note the types of R-groups they carry. Next, let's create a new 2D view in DataWarrior, and in this plot we'll place R1 on the x-axis and R2 on the y-axis to visualize how different combinations of substituents are distributed. Then open the Configure View menu, go to Marker Color, and choose to color according to p-values. This way, highly active compounds will stand out visually, making it easier to spot SAR trends. At present, we focus on the R1 column. We want to sort the group following our rules, not the order in the file. On the column name, do one click with Control key, or on a Mac, do right mouse click and then use Set Category Custom Order. You can use the parameters shown in this slide and click OK. From this figure, it seems that a relatively short R2 group is important. The molecules are colored according to p-values, but it is also possible to show these values on the plot. Just use the Configure View command and set marker labels and select p-values. It is then easier to see and decide where to focus. Now we plot R1 on the x-axis and R3 on the y-axis to visualize the molecules. And we look at the best two compounds. The top compound according to p-value, we call it compound A, and the second one we call it compound B. Of course, here it is to test the commands. There is no major differences between the two compounds. We could imagine to design a compound not present here by changing some R groups. This is what we are going to try. We imagine a new compound with the scaffold shown that is present in compounds A and B, and then the R1 of compound B and the R3 of compound A. We do not change the R2 group as present in the two molecules. At this point, we'll keep all our molecules in the dataset, but focus on the most active compound. To do this, simply sort the dataset by p-value and select the compound with the highest activity. In our case, we will call it compound A. Then, we'll create a new window in DataWarrior dedicated to compound A, as shown on the screen. This is the result. We have now a new window created with only compound A. We will then modify this compound here without losing our original molecules. Then we go back to the main window, right mouse click on the R1 chemical group of compound B and copy the 2D structure as shown on the screen. We then move to the new window where we have compound A alone. We edit the cell of the R1 group, use the lasso tool, delete the original R1 and paste the R1 of compound B. In this new window, we can go to the menu, Data, and select Merge Columns as shown. Then we select the 4 columns we want to merge and click OK. The previous commands will create a new column named here Merge Data, and you see a new molecule created there, a mixture of compound A and compound B. Now that we have this new molecule, we can predict some properties, we can generate a 3D structure, and so on. We can also copy the 2D structure and search if this new molecule is present in the ChEMBL database. Now let's see how we can use DataWarrior again to search for similar molecules in the ChEMBL database. To do this, go to Search Database and select ChEMBL. Then choose Similarity Search and paste the 2D structure of our newly created molecule into the Structure Editor window. We'll leave the other parameters at their default settings. Again, this is just an exercise, but it demonstrates how DataWarrior can connect your design directly to publicly available bioactivity data and help you explore structurally related compounds. After running the similarity search, we retrieve about 207 molecules from the database. Now, we can check whether our newly designed molecule is already present in the database or if it's potentially novel. We can also explore the biological targets these similar compounds bind to, their reported activity values, and any associated pharmacological data. This step gives us a better understanding of where our design molecule fits in the known chemical space and can help us identify promising directions for optimization. Drug discovery is an incredibly challenging journey, and what we've explored today through core-based SAR analysis represents just a small slice of why this field is so complex. Even with powerful tools like DataWarrior and vast open databases, understanding how structural changes translate into biological activity remains a central challenge. But mastering these techniques, from scaffold analysis to R-group exploration and CR-driven design, gives us the tools to make smarter decisions and design better compounds. That's it for today. Thanks for following along, and I hope this introductory tutorial has helped you appreciate how data-driven SAR can guide innovation in drug discovery. I'll be sharing more tutorials as we continue exploring DataWarrior and its powerful features, so stay tuned. And until next time, take care.